Yeah, this one. In the 1940s, biochemist Erwin Chargaff determined that the amount of A in a DNA molecule equal the amount of T, and the amount of C equal the amount of G. This finding was called the Chargaff's rule. So, with mathematical equation, it is simple. Yeah, the amount of A is always the same with the amount of T, and the amount of C is equal with the amount of G. Nah, so purine ya over pyrimidine ya, the ratio will always close to 1. Yeah, because purine is G plus A, right? And then pyrimidine is C plus T. Since A equals to T and C equals to G, then purine over pyrimidine equals to 1. Yeah.
So purine is A and T and
Purine is A and G.
Ah.
All right here. Okay. So purine is A and G. And then pyrimidine is C plus T. This one. Okay.
Yes.
Yeah. Okay, we move to the next slide then. Nah, DNA is a double helix. Nah, the invention of DNA is double helix occur in later time. Yeah. To be precise, it is in the 1940s. Several other scientists investigated the structure of DNA. Yeah. Rosalind Franklin and Maurice Wilkins studied the structure of DNA crystals using X-ray diffraction. So X-ray diffraction is a technique, yeah, that they use at that time because in the 1940s we don't have Twitter, we don't have Instagram, and we don't have any sophisticated tools like cryo-EM microscopy. What we have back then is this one. This is the most sophisticated that we have in 1940s. We call it as X-ray diffraction. So when you go to hospital to examine your skull, yeah, your bones, you use this X-ray machine. Yeah, I believe you already familiar with that. But in this experiment, we will use the X-ray machine to examine the crystal of nucleic acid or the crystals of DNA. You shoot the X-ray and then we will examine the diffraction pattern that is formed onto the film. Yeah. Now, from that diffraction pattern, of course, by using mathematical equation and analysis, yeah, they can deduce, yeah, several qualities of DNA. First, they deduce that it is long and thin and has a uniform diameter of 2 nanometers. 1 nanometer is 10 over, 10 power to minus 9. So 10 power to minus 9 meter. So it is very, very, very small. We cannot see it with naked eye. When you want to see this type of molecule, you should use a special equipment, yeah, called as atomic force tunneling microscope or something. I forgot the exact name, but it can dissect up to nanometer scale. And then the second point that they made that DNA is helical, that is twisted like a corkscrew and contains and consisting of repeating subunits. Yeah. Nah, this is the diffraction pattern, just like this one. Yeah. So it is a tedious work. Indeed, it is. Yeah. To elucidating this one. Yeah. This diffraction pattern of DNA result that you get to construct the double helix model of DNA. Now, this is the scientist we call him Maurice Wilkins, and this one is Rosalind Franklin. Yeah. So this is also quite challenging, the story of women in science. Later on, I'll show you the video, the TED video. I'll send it to your WhatsApp. If you have spare time, you can watch how actually Rosalind Franklin contributes, yeah, to this very important body of work. But sadly, Rosalind Franklin already passed away due to ovarian cancer, maybe due to the exposure of radiation from the X-ray machine itself. Because at that time, when you work at the lab, people at that time do not really put attention into the safety precaution that they should pay attention to when they conduct the experiment. So it takes a toll on her body, and one of it manifested as ovarian cancer. And then she died at a pretty young age, I think 40 or something. And Nobel Prize, it's like an Oscar prize for science. They don't give any uh, what do you call it, post-axiom award. Yeah, so there is no post-axiom award. For example, if a scientist already died when the Nobel Prize is awarded, it cannot be awarded. Yeah, so that's the sad thing about Nobel, and scientists already make what do you call it, a proposal to change on this rule. Yeah, because it's nonsense, right? It's doesn't make sense, illogical. Now, so that's why if you examine the Nobel website, the scientist that got the recognition with Nobel for the invention of the structure of DNA is Maurice Wilkins, Jim Watson, and Crick, which those three scientists are all males. Yeah, so you can see how at that time science was a very man-centric field. Yeah, and women were marginalized. But nowadays the situation is pretty different, especially in developed countries. Yeah, you can see that mostly scientists are usually women, especially in biology, in chemistry. The latest one, the Nobel Prize in Chemistry, it is Jennifer Doudna and Emmanuel Charpentier. Yeah, which both of them are women. Yeah, the first time. Yeah. Okay, so you can delve further into the story later on. This one, James Watson and Francis Crick combined the X-ray data with bonding theory to deduce the structure of DNA. But what they did is actually they see it on purpose, yeah, not accidentally, yeah, but on purpose. They borrow Franklin data, yeah, from Wilkins without telling or without asking permission from Rosalind Franklin. So actually, it's like a bit of stealing, quote unquote, yeah, stealing Rosalind Franklin data. So they publish at the same time, but the journal put the James Watson and Francis Crick publication in the front page, and then after that, Rosalind Franklin's at the next page. So it's pretty bleak and sad situation at that time. Yeah, because actually the one who made the work, yeah, the important work that go to the lab and then do the tedious process of shooting the crystal with X-ray machine is Franklin. Yeah, Watson and Crick just making the model, but the one who are obtaining the raw data, yeah, the data ownership is actually it is Rosalind Franklin. Now, from that one, we know that DNA is made of two strands of nucleotides. Within each DNA strand, the phosphate group of one nucleotide bonds to the sugar of the next nucleotide in the same strand. The deoxyribose and phosphate portions make up the sugar phosphate backbone. I'll show you the picture. This is the model that Watson and Crick made. Yeah. So the nucleotide bases protrude from this backbone. All the nucleotides within a single DNA strand are oriented in the same direction and thus have an unbonded sugar at one end and an unbonded phosphate at the other end. The hydrogen bonds between complementary bases, hold the two DNA strands together in a double helix. The bases protrude inward toward each other from the sugar-phosphate backbone like rungs on a ladder. So the rungs of the ladder is the nitrogenous base. And then the hydrogen bonds hold these base pairs together. The ladder-like structure is twisted into a double helix. Yeah, the ladder-like structure is twisted into a double helix. Okay. Hydrogen bonds between complementary bases hold two DNA strands together in a double helix. The two strands in a DNA double helix are said to be antiparallel. Yeah, what is antiparallel? It is oriented in opposite direction. Later on, I'll show you the picture. From one end of the DNA molecule, if one strand starts with the free sugar and ends with the free phosphate, the other strand starts with the free phosphate and ends with the free sugar. Yeah. Nah, this one. So free phosphate to free sugar. Yeah. And then this one, free sugar to free phosphate. So the orientation is said to be anti-parallel. Yeah. And then you can see the hydrogen bonds in the middle here, denoted by the dash, red dash here. A and T is connected by two, only two, hydrogen bond. While C and G, not two, but three hydrogen bond. So the number of hydrogen bond is different between A and T to C and G. Yeah, it is 2 versus 3. Okay. Now this one. Adenine bonds only with thymine, cannot with the other. Yeah. So the bonding is precise. Yeah. The principle of complementary base pairing. And guanine or G bonds only with cytosine or C. Bases that bond with each other are called complementary base pairs. Thus, if one strand has the base sequence CGT, TTAGCCC, the other strand must have the sequence GCA, AAT, CGGG. Yeah. So this is just an example. Later on, we will have more practices. Nah, therefore, this complementary base pairing, yeah, explains Chargaff's law, Chargaff's rule that we have discussed previously, that for a given molecule of DNA, adenine equals thymine and guanine equals cytosines. Since every adenine, for example, is paired with a thymine, no matter how many adenine are in the DNA molecule, there will be an equal number of thymines so that they can pair up. Okay? Now, adenine and guanine are large molecules since they are double ring. So in terms of its molecular size, they are larger compared to thymine and cytosine that are relatively smaller. Because base pairing always places a large molecule with a small one, the diameter of the double helix remains constant. In 1953, James Watson and Francis Crick consolidated all the historical data about DNA into an accurate model of its structure. Now this one. Yeah, this is the one that is usually depicted in any biology textbook. Yeah. Now, how can a molecule with only four simple parts be the carrier of genetic information? Yeah, transmission of information is key here. The key lies in its sequence. Yeah, the enormous probability that can be resulted only from those four letters. Yeah, and not the number of subunits. Within a DNA strand, the four types of bases can be arranged in any linear order, and the sequence is what encodes genetic information. Yeah. Now, the genetic code ATCG is analogous to languages. We have A, B, C, D, E, F, G, H, I, J, K, L, M, N, O, P, up until Z, where small sets of letters combine in various ways to make up many different words. English has 26 letters. Yeah, same with Bahasa Indonesia also. Hawaiian has 12 letters. And in computer science, the binary language of computers uses only two letters, 0 and 1. Yeah, or on and off. In biology, we have four. Now, the sequence of only four nucleotides can produce many different combinations. A 10 nucleotide sequence, for example, can code for more than 1 million different combinations of the four bases just from the 10 nucleotide sequence. So you can imagine if it is 20 nucleotide sequence, 100 nucleotide sequence, of course, the resulting combination will be much a lot more than this. Okay. Now, how does DNA replication ensure genetic constancy during cell division? Now, this part talking about the next one, which is the DNA replication process. So up until this point, the discussion of DNA structure will be finished. And then we will move to the next part of our lesson, yeah, which is DNA replication. So replication of DNA is a critical event in a cell's life. Why? Because omnicellulae et cellulae. That means all cells come from pre-existing cells. So cells reproduce by dividing in half. Yeah, there are mitosis. Yeah, one of the cell division, and then cell division type, and then each of two daughter cells gets an exact copy of the parent cell's genetic information. Yeah, how come? Because there is duplication of the parent cell DNA that we call as DNA replication. Yeah. Now, DNA replication produces two DNA double helices, each with one original strand and one new strand. The fact of complementary base pairing suggests a model for how DNA replicates. The ingredient for DNA replication are threefold. Yeah, you have three ingredients. First, the parental DNA strand should be there. Second, the free nucleotides. The free nucleotides means the raw material, yeah, that you need to make the new DNA molecule. And then the third one, do not forget the enzyme, because enzyme hold very vital role in life. So a variety of enzymes mean not just one enzyme, but many that will unwind the parental DNA double helix, and then some of them will synthesize the new DNA strands. Yeah. Nah, so we go into further detail now. DNA replication has objective to produce two DNA double helices, each with one original strand and one new strand. Yeah, there are several types of enzymes that involved in this process. The first one, it begins with enzyme called DNA helicases that pull apart the parental DNA double helix at the hydrogen bonds between the complementary base pair. And then the second one, a second strand of new DNA that is synthesized along its separated strand by DNA polymerases, which pair free nucleotides with their complementary nucleotides on each separated strand. Ya. Nah, the DNA replication produces two DNA double helices, each with one original strand and one new strand. When replication is complete, its parental strand and the daughter strand that was copied from it by base pairing wind together into a new DNA double helix, thus creating two copies of the original double helix. I'll show you the picture. Now this one. Can we play the animation first? Oh, the animation cannot be played. So later on, I'll give you the uh link to the YouTube video. You can watch it after this lesson end. Okay, Leah? Okay. Now we will move to the next one. Now, this is the diagrammatic representation. Yeah, the basic features of DNA replication. The first, you have the parental DNA double helix. The second, this parental DNA should be unwound so that the new DNA strand can now be synthesized with bases complementary to the parental strands. So the free nucleotides will join in here. Yeah, of course, with the help of a certain enzyme called DNA polymerase. Yeah, up until the end. So now its new double helix is completed. Yeah, and composed of one parental strand. Yeah, the blue one, and one new strands in red. Yeah. So this is the semi-conservative model of DNA replication. Now, DNA replication produces two DNA double helices, each with one original strand and one new strand. Base pairing is the foundation of DNA replication. An adenine on one strand, yeah, pairs with a thymine on the other strand, A with T, C with G. So if a parental strand reads TAG, the new strand reads A, T, C, according to the base pairing principle. So you should keep in mind about this one. Don't forget about this one. A, T,
Uh,
C, G. Any question?
Can you go back?
Okay. This one.
Uh, mm, okay.
Okay. Now we move to the next one. DNA replication produces two DNA double helices. It's with one original strand and one new strand. The two resulting DNA molecules have one old, sorry, have one old parental strand and one new strand. We call this replication as the conservative replication. Yeah. So semi-conservative replication of DNA means that from one DNA double helix molecule, it will produce two identical DNA double helices. Yeah. Each with one parental strand and
Mm.
how to ini ya? Wait a minute. Each with one parental strand, the blue one, and one new strand, the red one. Accurate replication and proofreading produce almost error-free DNA. Infrequent changes in the sequence of bases in DNA result in defective genes called mutations. Mutations range from changes in single nucleotides to movements of large pieces of chromosomes. Mutations may have varying effects on function. Yeah. So not only when we want to make books, you have editor to proofread everything. The typo, the misspelling, and so does the DNA replication. They have editor to check that the DNA is almost error-free. What does it mean? The word almost means there is still infrequent changes in the sequence of bases in DNA. We call this as mutation, yeah, spontaneous mutation, ranging from changes just in single nucleotide, so just one letter, to movement of large pieces of chromosome, yeah, hundreds of letters, thousands of letters. Therefore, the effects also comes in varying degree, yeah. Some is beneficial, but frequently mutation usually has deleterious effect, rarely beneficial. Yeah. Nah, during replication, DNA polymerase enzyme mismatches nucleotide once every 1,000 to 100,000 base pairs. Yet completed DNA strands contain only about one mistake in every 100 million. Imagine, amazing, right? To 1 billion base pair. So only one in every 100 million. In human, it means that this amounts to less than one error per chromosome per replication. This reduction in errors is accomplished by DNA repair enzyme, which proofread. Nah, so this is the editor, yeah, DNA repair enzyme, which proofread its new daughter strand and replace mismatched nucleotides. Proofreading occurs both during and after replication. Yeah, mistakes do happen. DNA is altered or damaged in a number of ways. Mistakes are made during normal DNA replication. Certain chemicals, for example, some compounds from cigarette smoke, increase DNA error during and after replication. UV radiation, that's why you should use sunblock, lotion, right? When you go outside and you go to the beach to protect you from this type of radiation that can contribute to incorrect base pairing that may result in skin cancer. Up until this point, any question?
Or can you hear me?
Yes, I can hear you.
I cannot hear you.
Okay. Test, test. Hello, hello.
Oh, okay, yes. Now.
Yeah, okay. Okay. Now, mutation range from changes in single nucleotide pairs to movement of large pieces of chromosome. Yeah. So there are three types of mutation. The first one, point mutation, also called nucleotide substitution, involves the changes to individual letters, individual nucleotides in the DNA sequence. Yeah. So, for example, one type of point mutation occurs when a repair enzyme finds a mismatch but mistakenly cuts out the correct base and puts in the complement of the erroneous base. And then the second type, we have the insertion mutation. that occurs when one or more new nucleotide pairs are inserted into the DNA double helix. And then the third one, you have deletion mutation that occur when one or more nucleotide pairs are removed from the double helix. So insertion means you insert it, and then deletion means removing, and then point mutation can be substitution, yeah, one letter with the other letter. Okay. Now, and then other type of mutation. Inversion occurs when a piece of DNA is cut out of a chromosome, turned around, and reinserted into the gap. And then the second type of mutation is called translocation. Occurs when a chunk of DNA, often very large, yeah, is removed from one chromosome and attached to another, and it can be in flip position. Yeah, for example, 1, 2, 3, and now become 3, 2, 1 because now it is flipped. Now, in pictures, this is like this. Nucleotide pair change from AT, this one, to TA. Nah, this is called a substitution. And then next one, insertion. So the original DNA sequence 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 letters. So there are 12 pairs of letters. Now it became 13 pair of letters because one extra is now inserted, the TA nucleotide pair. This is called as insertion mutation. Next one, deletion. The original you have 12 pairs of letter. Now it becomes only 11 because CG nucleotide pair is now deleted. Yeah, it's now removed. This one. Yeah, so now the CG is only one. This one is double CG right here and here. Now you only have one. So that is called as deletion mutation. Next one, inversion. Original DNA sequence that undergo breaks at a certain point and it flips. So after the flipping, the DNA segment is inverted. We call it as inversion. Yeah, like you rotate a picture, yeah, and then you put that picture back to the original one. Translocation. Nah, DNA segments switch, yeah, not only break, but after the break, yeah, usually involving more than one DNA strand. In this picture, there are two DNA strands involved, the top strand and the bottom strand. Both of those strands break at a certain point, and then after that, they switch to one another. So now the top strand becomes longer, and then the bottom strand from long becomes short, as you can see in the picture. Okay? Mm-hmm. Next one. Nah, mutation may have varying effects on function. Mutations are often harmful, and an organism inheriting them may quickly die. As I already said to you before, usually the effect is deleterious. Some mutation may have no functional effect, or we call it as silent mutation. But some may be beneficial, but this is rarely the case, and provide an advantage to the organism in certain environment. Now, these advantageous mutations may be favored by natural selection and are the basis for evolution of life on Earth. Yeah, this advantageous mutation may be favored by natural selection and are the basis for evolution of life on Earth. We will learn about evolution in chapter 4, if I'm not mistaken. That's why you should learn about cell first, about biochemistry first as the fundamental, and then after that you can move to the next chapter, physiology, and then evolution, developmental biology, and so on. Wait. I think the slide is over. Already 51. Nah, so this one I already sent it to your email. You can check it and open it from your own laptop later on. And I'll send some video and also screenshot, yeah, from this meeting. After this, you can work on the question. And then if you already work on the question, let me know so that we can discuss the answer.
Mm.
Have you finished up until what number?
Now?
You can start to work now. We still have 10 minutes, right? It's already 80 minutes past. So you have 10 minutes. I think number 1 to number 10 for the first. And then we will go to the next meeting for number 11 and the next question. Yeah. Okay. I'll stand by here. If you already finished with those 10 numbers, just let me know so that we can discuss.
Okay.